Hello, my name is Serra, and I'm going to present an investigation of porous PCL scaffolds in osteogenic and angiogenic inducing microRNAs for bone tissue engineering. Bone is highly vascularized tissue made up mostly of collagen and calcium phosphate. Bones serve as the body's structural framework, protect internal organs, facilitate movement, store minerals, and produce blood cells. Trauma, infection, fracture, tumor resection disease, or congenital abnormalities can cause bone defects. Small defects can regenerate naturally, while larger defects and those in poorly vascularized areas require intervention. Autografts are the gold standard for bone regeneration, but they come with drawbacks such as pain at the donor site, multiple surgeries, infection risk, and limited graft availability. Bone tissue engineering focuses on designing materials that outperform and overcome the limitations of autografts by using the vertebrae's own repair mechanisms to repair the defect. Bone tissue engineering combines multipotent cells, scaffolds, and bioactive factors to induce osteogenic differentiation and proliferation. Mesenchymal stem cells were used for this study because they have the ability to differentiate into bone-forming cells, and human umbilical vein endothelial cells are associated with angiogenesis. Scaffolds are 3D structures designed to mimic the extracellular matrix. They provide a framework for cells to attach, grow, and differentiate. Our scaffolds were made from PCL, which is a synthetic polymer that we use for mechanical strength. Scaffolds are created with a porous structure. Pores allow nutrients, oxygen, and waste products to move in and out of the scaffold. Scaffolds are coated with gelatin, which is derived from collagen, a major protein in the extracellular matrix. Others are coated with hyaluronic acid, which is a naturally occurring molecule found in the ECM. MicroRNAs are our bioactive factors. They are small RNA molecules that play a crucial role in regulating gene expression. MicroRNA 26A has been shown to induce bone regeneration by regulating osteogenesis and angiogenesis coupling. Our objectives are to construct a composite, porous biomaterial system that supports MSC and human umbilical vein endothelial cell attachment and proliferation, and to evaluate the osteogenic and angiogenic inducing properties of microRNA 26A. We hypothesized that variations in scaffold porosity, origin size, and coating would result on differing effects of proliferation on MSCs and HUVECs. The three variants of microRNA 26A would exhibit different degrees of effectiveness in inducing osteogenic and angiogenic properties in MSCs. Fabrication of PCL scaffolds. We dissolved PCL in DCM, added NaCl, poured this into a petri dish, and allowed to dry in a fume hood overnight. Then we punched out discs 8 micrometers in diameter by 1 micrometer in height. Salt was leached from the scaffolds using the solvent casting method, and the scaffolds were lyophilized. Cellularity study for MSCs and HUVECs. Culture MSCs and HUVECs, coat scaffolds with either gelatin, hyaluronic acid, or media, seed the scaffolds with MSCs or HUVECs, prepare MTTSA, and measure absorbance at 570 nanometers in a spectrophotometer. Transfection and analysis of microRNAs in MSCs. Osteogenic and angiogenic inducing microRNA 26A variants were selected through literature research. MSCs were cultured, cells were transfected with microRNA using lipofectamine. 7 days after transfection, our mRNA levels were measured by RT-qPCR, and 7 days after transfection, protein levels were determined by immunoblotting. Initial HUVEC and MSC attachment was unaffected by porosity, portion size, or coating. Small pores limited cell migration, where large pores diminished surface area cell adhesion. Surface area is important for cell attachment during seeding, but cell migration is improved in scaffolds with pore sizes that are above 300 micrometers, which overrides the higher initial cell attachment seen with smaller pores. Coating was important for HUVEC cells and not for MSCs. MSCs transfected with microRNA 26,5P and 13P expressed higher protein levels of frunx compared to negative control. MSCs transfected with microRNA 26,13P expressed higher protein levels of VEGF compared to negative control. MSCs transfected with all three microRNAs expressed higher protein levels of angiopoietin compared to negative control. All three microRNAs resulted in significantly increased mRNA levels of RUNX2 and VEGF-A. Transfection with microRNA 26A, 13P, and 23P induced higher ALPL and BGLAP mRNA levels. 26A, 3P had the highest expression of osteogenic and angiogenic markers tested. We conclude that optimal scaffolds for HUVEC and MSCs can be fabricated with 80% porosity, portion size range between 300 and 500 micrometers, and coated with gelatin. The study revealed microRNA 26A variants can promote osteogenesis and angiogenesis in MSCs by upregulating markers associated with these pathways. 13P and 23P had higher effectiveness compared to 5P. The results from the study contribute to our overall goal of developing a composite porous biomaterial system seeded with MSCs expressing microRNAs that can induce osteogenesis for bone tissue engineering.